You're now focusing on the spinal cord in its full three-dimensional and sectional perspectives, using both model-based and digital tools like slides and the Anatomage table. Let's explore how each layer, tract, and landmark integrates into the whole structure, starting from external identification to cross-sectional detail. Begin by observing the full-length spinal cord on the Anatomage table. From the foramen magnum at the base of the skull, the spinal cord descends down through the vertebral canal and ends around the level of L1 or L2 in adults. Below that level, you can see the cauda equina, a bundle of descending spinal nerve roots. The cord itself is roughly cylindrical, about 42 to 45 centimeters in adults, and is segmented into cervical, thoracic, lumbar, sacral, and coccygeal regions. As you zoom in on the cervical region, note the cervical enlargement. This is more prominent due to the increased number of motor neurons innervating the upper limbs. Similarly, in the lower lumbar region, you'll identify the lumbar enlargement, related to lower limb innervation. Now rotate the model to view the anterior surface. You'll notice a deep longitudinal groove called the anterior median fissure. This groove marks the midline and helps in orientation. Just lateral to it, the ventral roots emerge from the spinal cord. These contain efferent motor fibers. Switching to the posterior surface, observe the shallower posterior median sulcus. Lateral to this sulcus, you'll find the dorsal rootlets converging to form the dorsal roots, which contain afferent sensory fibers. The dorsal root ganglia, visible as bulges along these roots, house the cell bodies of sensory neurons. Look closely at a thoracic level cross-section on slide images. The internal anatomy becomes clearer. The spinal cord's gray matter sits centrally and is shaped like a butterfly or the letter H. This gray matter is divided into dorsal, posterior, horns, ventral, anterior, horns, and, in the thoracic region, lateral horns. The dorsal horns receive sensory information from the periphery, while the ventral horns contain the cell bodies of lower motor neurons. Between the right and left halves of gray matter lies the gray commissure, traversed by the central canal, which is lined with ependymal cells and contains cerebrospinal fluid. Surrounding the gray matter is the white matter, divided into anterior, lateral, and posterior funiculi. Each of these contains ascending and descending tracts. The posterior funiculus contains the fasciculus gracilis and fasciculus cuneatus, responsible for transmitting fine touch, proprioception, and vibration. The lateral funiculus houses both ascending tracts like the spinothalamic and spinocerebellar pathways and descending ones like the lateral corticospinal tract. The anterior funiculus includes the anterior corticospinal tract and parts of the spinothalamic tract. Now shift to the Anatomage interface and highlight individual tracts. Activate the corticospinal tract overlay. You'll see it originating in the cerebral cortex, descending through the internal capsule, brainstem, and then into the spinal cord's lateral funiculus, where it synapses with lower motor neurons. Similarly, trace the dorsal column medial lemniscus pathway. Touch receptors from the lower limbs send signals via the fasciculus gracilis, and from the upper limbs via the fasciculus cuneatus. These axons ascend ipsilaterally and synapse in the medulla, before crossing over to form the medial lemniscus and ascend to the thalamus. Examine the meninges using the spinal cord model. The dura mater, arachnoid mater, and pia mater form three protective layers around the cord. The epidural space contains fat and venous plexuses. The subarachnoid space, between the arachnoid and pia mater, contains cerebrospinal fluid and extends down to S2, making it a safe site for lumbar puncture. Now identify the segmental levels using the spinal nerves. In a dissected model, trace a spinal nerve emerging from the intervertebral foramen. It is formed by the union of the dorsal and ventral roots. Each spinal nerve divides into a dorsal ramus, serving the back, and a ventral ramus, which contributes to plexuses supplying the limbs and anterior body wall. In summary, the spinal cord is highly organized, with clearly defined external landmarks like the anterior median fissure and dorsal sulcus, and internal features like gray and white matter subdivisions, tracts, and neuronal pools. The use of models, slides, and digital platforms like Anatomage brings this structure to life, revealing the spinal cord not as a passive conduit but as an active, dynamic integration center for sensory and motor activity.